Now you know about some of the specific pathogens that can cause diarrheal disease, and that most of these pathogens originate in human or animal feces. Well, there are so many different pathogens, it's impractical to try and measure them all. So what we do instead is we use indicator species to indicate the presence of fecal contamination. Today we'll talk about those indicator species and also look at a tool for charting out pathways of transmission of diseases called the F diagram. Finally, we'll touch briefly on chemical water quality, though it's not a focus of this course. It would be impractical and impossible, actually, to directly measure all the different pathogens that could cause diarrheal disease. So instead, we look for indicator species that can show the presence of fecal contamination. Some characteristics of an ideal fecal indicator are that it should be something that's universally present in the feces of both human and animals in large numbers. Those numbers should be higher than the actual numbers of the pathogens of interest. And the indicator should not be harmful or pathogenic in itself. It should be something that's readily detected using simple, inexpensive methods. And it should also persist in the environment or water in a similar manner to fecal pathogens. You wouldn't want it to actually grow in natural waters because then you would get misrepresentation. And finally, if possible, the indicator should respond to treatment in a similar fashion to fecal pathogens. One of the most common indicator species is a bacteria class called total coliforms. Total coliforms are bacteria that can ferment lactose and produce acid and gas at 35 degrees Celsius. There are many different families of coliforms: Citrobacter, Klebsiella, Hafnia, and Enterobacter. And for this reason, total coliforms is not really useful as a fecal indicator because many of these bacteria have non-fecal sources. However, total coliforms can be useful for operational monitoring to demonstrate, for example, the effectiveness of treatment or overall cleanliness and integrity in a distribution system. Thermotolerant coliforms, as their name implies, can tolerate heat, and these are a subset of the total coliform class. Thermotolerant coliforms are what used to be called faecal coliforms, and they can ferment lactose within 48 hours at 44 degrees Celsius. Feces contain a large number of thermotolerant coliforms, but there are some indications of non-fecal sources of thermotolerant coliforms, such as Klebsiella and some species of Enterobacter. Also, there's evidence that thermotolerance, under some conditions, can grow or die off in water at ambient temperatures. So it's not a bad indicator, but it has some limitations. Escherichia coli, or E. coli, is one specific species of thermotolerant coliforms that is generally agreed to be the most suitable indicator of fecal contamination. There's some possibility that E. coli may grow in the environment in tropical settings, and it is only moderately persistent, so the absence of E. coli doesn't necessarily mean the absence of fecal contamination. However, on balance, considering all the different ideal characteristics of an indicator bacteria, E. coli is considered to be the best available option. Indicator bacteria are usually reported in terms of the number of counts or colonies in a 100 milliliter sample. Many drinking water standards and WHO guidelines are set to be no detectable E. coli or thermotolerant coliforms in a 100 ml sample. You can consider that water that has higher levels of indicator bacteria is probably more likely to have higher levels of fecal contamination, though there's not a very clear correlation between indicator bacteria and pathogens on the other hand. Still, water having very high levels of E. coli, more than 1,000 counts per 100 mils, is probably very risky for consumption. Water having no detectable E. coli in it, however, might not be completely safe because E. coli is relatively less persistent and robust in the environment compared to some pathogens. For this reason, it would be preferable to combine a measure of water quality, such as an E. coli measure, with some kind of assessment of the system safety like a sanitary inspection, which identifies a number of risk factors at a water source. Then by combining a water quality measure on the vertical axis and a water management measure on the horizontal axis, we could make a risk matrix and prioritize areas, water sources for interventions. While E. coli or thermotolerant coliforms are the most commonly used indicator species, other indicators might be more relevant for certain purposes. Viruses travel in the environment very differently than bacteria, so one might want to look at coliphage viruses that require a bacteria host to survive as an indicator for viruses. Fecal streptococci are more hardy, robust bacteria compared to E. coli, so they could persist longer in the environment and maybe be a better indicator of long-term fecal contamination. Traditionally, methods for enumerating fecal streptococci have been more complicated, but this is changing with the development of new methods. Finally, protozoa can be very resistant to disinfection, to heat, to chlorine, to ozone, so E. coli is not a good indicator for treatment efficacy. But some bacteria, like Clostridium perfringens or some Bacillus species, can form spores which are roughly similar in size and resistance to some protozoan pathogens. Now, the pathogens that we're concerned about, whether they're viruses, bacteria, or protozoa, they mostly originate in feces, but they don't cause health problems unless they're ingested by a new host, which means taking them in through the mouth, which is on their face. So we can think about how can pathogens travel then from feces to faces. And there's a useful learning tool for describing the different pathways. It's called the F diagram. Here's a nice picture of the F diagram developed by Weddick in Loughborough, the United Kingdom. And it's called the F diagram because many of the intermediate phases in the environment where contamination can take place have words that start with the letter F. So if we start, of course, with feces, how can we get to faces? Well, we can go through fluids, which is the subject of this course. And actually, the nice thing about this diagram is you can see with a blue star where the water-related pathways are. So between feces and fluids, you have water source protection. And after the fluids to the host, you have transport, storage, and treatment of water. Besides the fluids, you have fingers, which can directly contaminate fluids or foods or faces, and also flies. Remember the trachoma, which is caused by flies landing on faces. Fields, here having poor sanitation is a major risk factor. And flooding, again, if there's poor sanitation, can lead to exposure to pathogens. Now let's focus in on those pathways between feces and faces that involve water. We can think about a safe water chain with different stages where contamination could be introduced. Contamination could occur at the water resource itself or during delivery to the consumer. Fecal matter can also be introduced during the collection and transport of water if the source is outside the home. When water is stored inside the household, it could become contaminated. And finally, the drinking water vessel itself, a cup or a glass, might be dirty and introduce fecal matter. Now let's look at an illustration of these different stages in a safe water chain. First, the water resource itself can be contaminated from contact with human or animal feces. Surface water often contains fecal bacteria, but even groundwater aquifers can be contaminated by inadequately managed sanitation. Even if the water resource is clean, or if water is purified at a treatment plant, it can become contaminated during delivery to the consumer. For example, in a piped water supply, if there is not continuous pressure in the distribution system, contaminated water may enter the pipes through cracks or joints. Another example, though, could be an improperly constructed rising main on a borehole or suction hand pumps, which are primed with unsafe water. The point of collection itself can be contaminated. Think of a communal tap or borehole spout, which is handled and contacted by adults, children, and even animals. And when the point of collection is outside the home, people must use containers, which themselves may be dirty. And people often rinse containers and wipe them with their hands before filling them up. But if their hands are not clean, this may not help at all. The containers must then be transported back to the household, giving another opportunity for contamination. Open containers are especially vulnerable because it's easy to contact the water with hands during lifting and carrying, and also dust or insects can enter an uncovered container. Once the water is taken to the household, it is often transferred to another container and may be stored unhygienically in open containers or on the floor where children and animals can introduce pathogens. Point-of-use water treatment at this stage offers the possibility of inactivating pathogens introduced at earlier points in the water chain. But even after storage, contamination possibilities exist. Water may be retrieved from storage containers using an unclean dipper or ladle, especially if the storage container is uncovered. Finally, the drinking vessel itself, a cup or glass or perhaps a gourd, can be full of fecal contamination. Again, people may wipe a drinking vessel with their hands before using it, but this might actually make the glass dirtier. So there are many opportunities for contamination between the original source of drinking water and the actual point of consumption. A big advantage of household water treatment is that it offers the possibility of removing any contamination that is introduced at the water resource, the delivery system, or through collection and transport. Many household water treatments don't prevent recontamination, so it is important to have safe storage after a treatment step. And likewise, good hygiene, especially washing hands with soap, is important to minimize the possibility of clean drinking water becoming contaminated just before consumption by using a cup or glass that is fecally contaminated. So in this module, we've talked about different indicators of fecal contamination and how E. coli is the preferred indicator bacteria. We've shown you the F diagram, which illustrates different pathways between feces and faeces that can lead to exposure to pathogens, including through drinking water. And we've talked about different stages along the safe water chain from catchment to cup, where drinking water can become contaminated. Now in the next module, we'll introduce those treatment technologies for household water treatment and safe storage.